Let us have an overview of glycolysis. Remember, there are 10 steps that you need to be thorough with, and you have to remember the names of the 10 enzymes. First, you get the meaning of the term glycolysis. Glyco refers to glucose, and lysis refers to breaking or splitting. So in the cell cytoplasm, glucose gets broken down to two molecules of pyruvic acid, or you can simply call it as pyruvate. So one molecule of glucose always generates two molecules of pyruvate, resulting in the net generation of 2 ATP and 2 NADH. Remember, ATP and NADH are high energy carrier molecules. You should be aware of the location of the glycolysis. Remember, it happens in the cell cytosol, or you can simply call it a cell cytoplasm. This process do not require any oxygen, so it is known as anaerobic phase. And if the question asks why glycolysis is known as anaerobic phase, you can simply write it down that it do not require oxygen, right? And it consists of 3 phases, and you have to be thorough with the 3 phases of glycolysis. Number 1, it is known as energy consumption phase, which means there is a consumption of ATP. And second phase is splitting and rearrangement phase. And the third, it is known as energy payoff phase, meaning there is generation of ATP. So you have to be thorough with these 3 phases. So now let's get into the detail of each phase. In the first step of energy consumption phase, glucose gets phosphorylated, meaning there's the addition of phosphate onto glucose, and the phosphate we obtain it from ATP. So there is a consumption of ATP here. And when we say ATP consumption, we are actually breaking down ATP. So we are hydrolyzing ATP. The last bond between the 2 phosphate is broken, and when the bond gets broken, there is always release of energy, and this energy is used to form a bond between carbon and phosphate. So take this as glucose, and glucose has 6 carbons, right? Take this as first carbon, second carbon, third carbon, fourth, fifth, and sixth. So there are 6 carbons in total, and ATP provides energy. How do we obtain energy? By breaking bond between the last 2 phosphates. When the bond is broken, we separate phosphate from ATP, right? And we also obtain energy. And this energy is used to form a bond between phosphate and 6th carbon of the glucose, right? And in the meantime, ATP gets converted into ADP. So now it's no more glucose, it is glucose 6-phosphate. So there is a transformation of glucose into glucose 6-phosphate. And how did it got transformed? By phosphorylation. And the enzyme that catalyzes this reaction is hexokinase. Now, how do I remember the name of the enzyme hexokinase? I'll share a very simple technique here. Whenever ATP is associated in the particular step, I want you to take ATP as a kind soul. So take ATP as a kind soul. And you know that enzyme must end with ase, right? So keep it as kinase. So whenever there is ATP being associated, keep second name of the enzyme as kinase. And in this particular step, the first name of the enzyme, which is hexo, you can dedicate to the number of carbons found in glucose. So you can see there are 6 carbons, so 6 equals to hexo. So that's how you remember the enzyme name, hexokinase. And hexokinase is known as the initiator enzyme in glycolysis, without which glycolysis cannot be operated. And this is the first step of energy consumption phase. It mainly involves phosphorylation. And if you're wondering what phosphorylation is, it is simply addition of phosphate. And you can see there, phosphate is being added onto 6-carbon, right? So the technical term is phosphorylation. And remember who provides phosphate? The credit goes to ATP. Now in the second step of glycolysis, glucose 6-phosphate gets transformed into fructose 6-phosphate by the process called isomerization, which means glucose 6-phosphate simply undergo rearrangement process and it gets transformed into its isomer fructose 6-phosphate. And the enzyme that catalyzes the reaction is phosphoglucose isomerase. Now, how to remember the enzyme phosphoglucose isomerase? I will share this simple technique. Dedicate second name of the enzyme to process, so it will be kept as isomerase. And then the first name dedicate to the substrate. I am referring to the precursor substrate. Okay, precursor in this step is glucose 6-phosphate, and prioritize phosphate first, so phosphoglucose isomerase. That is the technique to remember the enzyme name. I repeat, second name dedicate to the process going on here, isomerization, so isomerase, dedicate to the precursor substrate, which is glucose 6-phosphate, prioritize phosphate, so it will be written as phosphoglucose isomerase. In the third step of glycolysis, fructose 6-phosphate undergo phosphorylation, meaning there is addition of phosphate. Take this as fructose 6-phosphate with 6 carbon atoms in it, which is denoted by number. There's already 1 phosphate attached onto 6 carbon, and when it undergo phosphorylation, that is addition of phosphate, we need phosphate, right? So we obtain it from ATP. ATP will generate 1 phosphate, and this phosphate will get attached onto first carbon of the fructose 6-phosphate. There is already phosphate attached onto 6 carbon of the fructose 6-phosphate, and the phosphate that we had obtained from ATP will get attached onto first carbon. So now fructose 6-phosphate will transform into fructose 1,6, and there are 2 phosphate in total, so bis phosphate. This will result in the transformation of fructose 6-phosphate to fructose 1,6-bis phosphate. And the enzyme that catalyzes this reaction is phosphofructokinase. Use the simple technique. Whenever there is ATP associated, keep the second name as kinase and dedicate first name to the substrate. Prioritize phosphate, so phosphofructokinase. Let us now recap the steps of energy consumption phase. In the first step, glucose undergo phosphorylation, which gets transformed into glucose 6-phosphate, and glucose 6-phosphate gets converted to fructose 6-phosphate, which is isomer, and fructose 6-phosphate gets transformed into fructose 1,6-bis phosphate with the phosphorylation. And when it undergoes phosphorylation, there is a consumption of ATP. ATP provides phosphate, which the phosphate gets attached onto the 6 carbon of the glucose, so therefore there is a generation of glucose 6-phosphate. And glucose 6-phosphate undergo isomerization, which generates fructose 6-phosphate. In the first step, the enzyme that catalyzes the reaction is hexokinase. Glucose 6-phosphate, when it undergoes rearrangement, it generates fructose 6-phosphate. And the enzyme that catalyzes this reaction is phosphoglucose isomerase. Remember the technique to remember the enzyme name. Fructose 6-phosphate, when it undergoes second phosphorylation, it utilizes ATP. ATP utilization generates phosphate, which gets attached onto the first carbon of the fructose 6-phosphate. So now it gets transformed into fructose 1,6-bis phosphate. The enzyme that catalyzes this reaction is phosphofructokinase. And this phase sums up the energy consumption phase. The next phase is known as splitting and rearrangement phase. Fructose 1,6-bis phosphate which has 6 carbon atoms in it. With the help of enzyme aldolase, remember it has 6 carbon atoms, so when it undergoes splitting, it will form 2 molecules which contains 3 carbon atoms in each. So there is distribution of equal number of carbon atoms, and the molecules are dihub and G3P. G3P can also be written as PGAL. G3P stands for glyceraldehyde 3-phosphate and dihub stands for dihydroxyacetone phosphate. Remember, they are isomer. So now there is a formation of 1G3P from the splitting of fructose 1,6-bisphosphate, and dihub being unstable, it will undergo isomerization, which means dihub will rearrange itself and it will get converted to another molecule of G3P. So now there is 1 G3P being produced from the splitting of fructose 1,6-bisphosphate, and there is another G3P being formed from the conversion of dihub. So now there is 2 molecules of G3P. And both G3P and dihub can be referred to as triose phosphate. So dedicate first name of the enzyme to them as triose phosphate and dedicate second name to the process. So you will keep it as isomerase, right? This is the technique to remember the enzyme name. The enzyme is known as triose phosphate isomerase, and this phase is known as splitting and rearrangement phase. Moving on to the third phase of glycolysis, which is energy payoff phase, meaning there is generation of ATP. It starts with the G3P dehydrogenation. So the 2 molecules of glyceraldehyde 3-phosphate, it undergo dehydrogenation. So the process is dehydrogenation, meaning removal of hydrogen. D refers to remove. So hydrogenation, there is a removal of hydrogen. And when the hydrogen gets removed, remember it always gets accepted by NAD+. So there is a removal of hydrogen from the substrate G3P. Since there are 2 molecules of G3P, 2 hydrogen gets removed. And the removed hydrogen gets accepted by 2 molecules of NAD+, which results in the generation of 2 molecules of NADH, which is a high energy carrier molecule. Phosphorylation will also take place in this step, which means G3P will get phosphate, but in this case not from ATP, it will get it from H3PO4. So H3PO4 will donate phosphate, which gets added onto G3P. So now G3P gets transformed into 1,3-bisphosphoglycerate or glyceric acid. Remember, G3P and 1,3-bisphosphoglycerate, they are not same molecule. 1,3-bis-PGA, which you can even write it as 1,3-bis-PGA, it is acid, and G3P is aldehyde. So the enzyme that catalyzes the reaction is G3P dehydrogenase. Dedicate second name of the enzyme to the process going on here, which is dehydrogenation, so keep it as dehydrogenase, and dedicate first name to the substrate, which is G3P. That's the technique to remember the enzyme name. So remember what's going on here. There is hydrogen being removed from G3P, which means there is dehydrogenation going on, and hydrogen which gets removed gets accepted by 2 molecules of NAD+, which results in the generation of 2 NADH, which is a high energy carrier molecule. Remember, there is also phosphorylation taking place here, and the phosphate, we obtain it from H3PO4. The phosphate gets added onto G3P. There is already 1 phosphate attached onto the 3rd carbon of G3P. And when another phosphate gets added onto G3P, it gets attached onto the 1st carbon, which results in the generation of 1,3-bis-PGA. In the next step, there is removal of phosphate from the 2 molecules of 1,3-bisphosphoglycerate. So there is a removal of phosphate from 1,3-bis-PGA. Remember, there is phosphate attached onto third and first carbon. And the phosphate which is attached onto the first carbon of 1,3-bisphosphoglycerate gets removed. And the removed phosphate gets accepted by ADP, which is adenosine diphosphate. Since there are two molecules of 1,3-bis-PGA, two phosphates are removed. And remember, these two phosphates are from the first carbon. And 2-phosphate gets added onto 2-ADP, which helps in the generation of 2-ATP. And that's how ATP gets generated in this step. This results in the generation of two molecules of 3-phosphoglycerate. Remember, one phosphate has been already removed from the first carbon, so there is no more 1 and no more bis now because there is only one phosphate, so you can't write the word bis, so it is only 3-phosphoglycerate. And the enzyme that catalyzes the reaction is phosphoglycerate kinase. Use the simple technique to recall the enzyme name. Whenever there is ATP association, the second name will be kinase, and the first name of the enzyme dedicated to the precursor substrate, which is phosphoglycerate. So therefore, it will be kept as phosphoglycerate kinase. In the next step, two molecules of 3-phosphoglycerate will simply undergo isomerization process, which means it will undergo rearrangement. And when it undergoes rearrangement, it will simply get transformed into its isomer, that is, two molecules of 2-phosphoglycerate. And the enzyme that catalyzes this reaction is phosphoglycerate mutase. In the next step, two molecules of 2-phosphoglycerate will simply undergo dehydration process. Since there are two molecules of 2-PGA, there will be removal of two molecules of water. And it will get transformed to two molecules of phosphoenol pyruvate. In short, you can write it as PEP, which is phosphoenol pyruvate. And the enzyme that catalyzes the reaction is enolase. Now, how do I remember the enzyme enolase? Use this simple technique. You see, we have been dealing with phosphate, we have been dealing with the glycerate, right? And now you're going to dedicate to the name that sounds unique. Phospho, it's common, right? Pyruvate, it's common. Glycerate, it's common. Which sounds unique? Enol. So, dedicate enzyme to the name that sounds unique, which is enol. So, keep it as enolase. That is the technique to remember the enzyme enolase. We are now with the last step of glycolysis. Two molecules of phosphoenol pyruvate, you can also write it as PEP. From PEP, there is a removal of phosphate. Since there are two molecules of PEP, you're going to remove two molecules of phosphate. So two molecules of phosphate gets removed. And phosphate which gets removed gets accepted by 2-ADP, which results in the generation of two molecules of ATP, and that's how ATP gets generated. If you want to generate ATP, remember to add phosphate to ADP. So this will ultimately result in the generation of two molecules of pyruvate, which you can also write it as pyruvic acid, in short PA. And the enzyme that catalyzes the reaction is pyruvate kinase. How to remember the enzyme name? Use this simple technique. Whenever there is association of ATP, give the second name as kinase and first name of the enzyme dedicate to the main mission of glycolysis, and the mission is to generate pyruvate, so keep it as pyruvate. And that's how you remember the enzyme name, pyruvate kinase. So now let us draw the complete flowchart of glycolysis. It starts with glucose, which gets converted to glucose 6-phosphate. Remember, phosphate, they got it from ATP. And glucose 6-phosphate gets converted to its isomer, fructose 6-phosphate, which undergo phosphorylation and generates fructose 1,6-bisphosphate. Again, remember, it got phosphate from ATP. Fructose 1,6-bisphosphate undergoes splitting into dihub and G3P. Remember, there are 2 molecules of G3P being produced. G3P can be written as PGAL. Now 2 molecules of G3P get converted to 2 molecules of 1,3-bis PGA, which gets converted to 2 molecules of 3-PGA, which is 3-phosphoglycerate, and 2 molecules of 3-PGA gets converted to 2 molecules of 2-phosphoglycerate, and 2 molecules of 2-phosphoglycerate gets converted to 2 molecules of phosphoenolpyruvate, which ultimately generates 2 molecules of pyruvic acid. Now, how do I remember the whole substrates of glycolysis? And you see, I know that you are struggling to remember the all 10 substrates of glycolysis. But don't worry, my dear. You can come up with silly things to remember important things. Remember, it's very difficult to remember important things, but easy to remember the silly things. That's how our brain works sometimes, right? So therefore, you can come up with silly things to remember the whole substrates of glycolysis. So you can use this technique. Start with glucose. Glucose. So use this silly song to remember all substrates of glycolysis. Glucose, fructose. Sing again. Glucose, fructose. Dihaplayasi pagal. Again, glucose, fructose. Dihaplayasi pagal. Pagal-layasi 1,3-bis PGA. 1 talk down, there's no more bis, so 3 PGA. 1 reduced down, 2 PGA. 2 PGA le pep pa. Again, glucose, fructose, dihaplayasi pagal, 1,3-bis PGA, 1 talk down, 3 PGA, 1 reduced down, 2 PGA pep pa. See, this silly thing can help you remember 10 substrates of glycolysis. To remember consumption of ATP and generation of ATP and NADH, use this technique. Start with ATP. In the next step, gane mindu. Dorong ATP, gane mindu, nadu. Since there are 2 molecules G3P, you're going to write NADH2. Start with ATP again. ATP, gane mindu, chudu. Dele ATP du. So again, ATP, gane mindu, okay, ATP, gane mindu, ATP, gane mindu, nadu, ATP, gane mindu, chudu, ATP du. So you can use this technique to remember ATP consumption and ATP and NADH generation in the steps of glycolysis. Regarding enzyme, whenever there is association of ATP, keep the second name as kinase and dedicate first name of the enzyme to the substrate. And next technique, dedicate second name to the process going on. And enzyme must end with ACE. So remember, sometimes we have to come up with silly things to remember important things, okay? Sometimes we have to come up with silly things to remember serious things. So, if you can't remember important thing, you can come up with a silly thing, create something, so that it helps you to recall easily. And that's the technique to remember the lesson for long term. To sum up, there are 3 phases of glycolysis. Starting from the glucose till fructose 1,6-bisphosphate, it is known as energy consumption phase. And fructose 1,6-bisphosphate getting splitted into dihub and G3P is splitting phase, and dihub getting converted into G3P is rearrangement phase. So together you can call it as splitting and rearrangement phase. And from G3P till pyruvate, it is known as energy payoff phase.